So now we've got those terms. Now let's look at those specific channels, okay? Those gates, if you will. All right. All of that resting membrane potential, all of that membrane potential in general, I guess I should say, is all due to our sodium and potassium channels. Okay. Some are gated to be stimulated only by a change in voltage. Okay. Some are not gated. They are always open. And these are usually referred to as leak channels. Okay. These are referred to as leak channels. Now, this book has leak channels as only being potassium. It depends on the textbook. In another previous textbook, it did refer to some sodium leaky channels. There are just a hell of a lot more potassium leak channels. So they outnumber sodium. So I'm going to just kind of keep with the concept of the book, because you'll be reading your notes and reading the book and listening to the lectures and looking at these notes over and over again. So I will keep it in terms of what this book states, okay? This book has potassium leak channels only. So let's just keep it as that, okay? Even though there are a few sodium leaky channels, the book doesn't make reference to them. So I'm just want to kind of keep it in jive with what the book has for your studying purposes here. So potassium have the leak channels, okay? No sodium leak channels. Voltage gated channels are only stimulated to open when the voltage changes. I have a picture on the next slide, but I'm going to draw a little bit of picture right here. Okay. Let's just say this is my length of axon. Doesn't matter. Axon, dendrite, plasma membrane of the soma, whatever. Let's just call it the axon for now. Let me change my pen color here. All right. Let's just look at sodium for right now. Okay. These guys are all sodium channels. No. Question? Okay. Okay. As an impulse, okay, as electricity is traveling down, okay, because it was stimulated by another sodium voltage gated channel here, right? So when electricity comes up to a voltage gated channel, it opens up and sodium is going to rush in, okay? I guess okay, this is the plasma membrane. This is my sodium gated channel that spans the entire width of it. And sodium rushes in from the extracellular into the intracellular, okay? As sodium rushes in, I am flip-flopping my charge. So what do I start to generate here? I start to generate electricity. And I get a change that occurs right here. And now this electricity starts to travel across. These voltage-gated channels are at the precise distance, right, that allows this electricity to travel this far before dissipating and stimulating another sodium voltage gated channel to allow sodium ions to come in, which causes a change in voltage here that allows electricity to be propagated along the length of the plasma membrane here. Stimulating the next one. Okay. Let me change my color real quick. Gray. Let's say, Okay. It didn't go that far. All right. For whatever reason, the stimulation was not significant enough, okay, to stimulate this from occurring. So none of this else happens. All right. So this stimulation has to be significant enough to travel this distance here.
There's a threshold that it has to meet, isn't there?
Yep. Okay. It has to travel this distance right here. Okay. This has to be enough to stimulate to be traveling across here, because depending upon the thickness, okay, depending upon how big this nerve is, right, depends upon, that will affect how fast that electricity can travel through the axon. All right. You're like, what the hell? Think of this visualization. Okay. Close your eyes. You are walking down the hallway on campus. Okay. Okay. It's not that wide. It's, maybe 8 feet wide. All right. If you had 100 people walking down that tiny 8-foot thing at one time, would it take you a long time to get down that hallway? Yeah, right?
Yes.
Yeah. Okay. You're bumping into people, it's slowing you down, it takes some time. If that hallway was 20 feet, that same number of people can walk through there a heck of a lot easier. Yeah. There's more space. I can quickly move through. I'm not going to be bumping in and interacting with anybody. I can easily breeze through that hallway. All right. That's basically the difference between a small nerve and a large nerve. All right. With a large nerve, there's more space. So that action potential can go down a little bit quicker because it's not bumping and interacting with stuff. When it's smaller, I'm bumping into more things, more ions, and that's going to slow it down. All right. So these voltage gated channels are interspersed at precise distances along the length of an axon. Not necessarily along the length of a dendrite. Okay? Not necessarily at precise locations along the surface of the nerve cell body. So if I don't have enough impulses coming into that dendrite, it may not be able to get in there and travel across the dendrite, travel across the nerve cell body, get to that axon hillock to get magic to happen. All right. So there are some very big things that can prevent an impulse from occurring on a nerve. One of the major things that gets magic to happen, that gets an impulse to occur, is the opening of these voltage gated channels. The great thing about these two type of channels, it is that depolarization event that gets sodium to open up and gets those sodium voltage gated channels to open up, gets sodium to come across. Once we reach plus 30 millivolts, potassium automatically opens up. There are certain phenomena in the body that things just happen because things just happen. We see this phenomena in many locations. One really important one is in the kidneys. Okay? When we look at a kidney, as you get deeper into the medulla, that it becomes more and more concentrated. Fascinatingly interesting as to why that is. But there are certain mechanisms in place to maintain that all the time. Same thing here. When it comes to a nerve, at plus 30 millivolts, potassium channels automatically open. Okay? Automatically open. And that helps us to get back to resting. If everything's at resting, everything's closed except for who? Leaky channels. Right? And generally, it's described in the vast majority of textbooks as sodium leak channels, sodium comes in, potassium leak channels, a potassium goes out. Right? and there's a constant movement across there because they're always open. And it's these leak channels that help to maintain this RMP. Okay. So we have leaky channels that are always open. We have voltage gated channels that only open when stimulated by certain phenomena. Sodium voltage gated channels when they're stimulated by an electrical current. Voltage gated channels generally are stimulated when they reach a certain membrane potential, plus 30 millivolts. Let's look at a picture of one, better than my drawing. Here is a sodium voltage gated channel. It is vastly different from a potassium channel. Why is it vastly different from a potassium channel? Because of one thing. Oh, get rid of yellow. The ball and chain. That ball and chain here. Potassium channel looks exactly the same as this, except that thing dangling off. That ball and chain there is what helps to allow sodium voltage gated channels to close as quickly as they do. Okay. This little ball and chain is referred to as an inactivation gate. Okay. When it comes to a sodium voltage gated channel, it has three states: closed, open, and inactivated. As you can kind of see here, protein channel is closed. Yes, would everybody agree with that picture there? Yeah. Okay, yep, can't get through there. Over here, when it's stimulated, it opens up. And as you can see that, there you go. It's opened up and there's my little channel where sodium rushes in. When we get to plus 30, it becomes inactivated. And it's referred to as an inactivated channel or an inactivated state. What does that mean? I have plugged the hole. I have plugged the hole. This little inactivation gate, and all it is just a protein chain here that comes up in like putting a stopper in a sink. Then it resets itself, closes, and once the closes, that little ball and chain goes back to its normal resting state.
So it's what allows the channel to close first. It stops the sodium from coming in, which then,
Yep.
which then lets it close fully. And then once it's done, the little ball goes back up.
Yes. Inactivation just refers to that ball and chain coming up and plugging the hole. Okay, that's all it does. Then it allows that sodium channel to reset itself. Okay. Potassium doesn't have that phenomena. And because it doesn't have that ball and chain there, that inactivation gate, we're just, okay. So would sodium be the exact same dang thing if it didn't have that inactivation gate? Yeah, it would. Okay, but because I have that inactivation gate, the ball and chain, okay, I plug the hole up. Now, can stuff come in here? No. Just like a stopper in a sink prevents water from getting down the drain, right? So that's how sodium channels, sodium voltage gated channels, are allowed to close a heck of a lot faster than a potassium would. When my sodium voltage gated channels open and it reaches that minimum threshold of minus 55, okay, that's threshold. So I have minus 70, I have minus 55. This impulse is going to be able to travel a significant distance, right? Because once you reach threshold, magic happens, right? That all or none type of response. But I have to get to threshold. I have to get to that minus 55 millivolts, or the impulse dissipates within the nerve. Okay? It's not enough to establish electricity to tickle and open that next sodium voltage gated channel. Okay? So I've got to reach threshold. Good gosh. So I've got to reach threshold. Again, which is minus 55. Obviously, when this sodium voltage gated channel open up, sodium rushes in due to that electrochemical gradient, positive attracted to negative, high on the outside, low on the inside. Okay. And it becomes deactivated at plus 30. So I got minus 70, I got minus 55, I've got plus 30. Those are the three numbers that you should remember when it comes to nerves, which will vary a little bit when it comes to muscles, but pretty close. I got minus 70 millivolts, my resting membrane potential. Threshold for a nerve is minus 55 millivolts. Peak is plus 30. It doesn't go above that. Okay. Deactivates at plus 30 millivolts. When I reach that plus 30 millivolts and I'm deactivated, that means sodium stops rushing across. And when sodium stops rushing across, the potassium voltage gated channel now opens up. Okay. When the voltage and when potassium voltage gated channels open up, as you can imagine, potassium rushes out and I repolarize. Everybody with me there on those voltage gated channels?